So here's for the agenda for the talk. I'm going to begin with some background on graph genome references. Then I will transition to talking about population models, bring the two subjects together with the generalization of a popular population genetic model to graph genomes. Then we've developed an algorithm for this model, and I'll show you some results. So let's start talking about graph genome references. And to motivate them, I have an example here of sort of a typical genomics pipeline where first you generate some sequencing reads, then you map the reads against your reference, and then you go on to do transcript quantification or variant calling, whatever. I want to focus on this middle step where we actually map the reads against the reference, and particularly on this guy down here, which is the reference sequence. I also want to draw your attention to this column here where all of the reads have a different sequence than the reference. And this is not a particularly uncommon or even interesting phenomenon. This is a homozygous alternative allele for a SNP, but it highlights a kind of simple and important point that even though we often call the reference sequence the human genome, it is not the genome of any human and never has been and never will be. Every sample will always have differences from it, and in this case, it didn't seem to have caused a lot of problems, but that's not always true. So here's a more severe example where the sample has a inversion relative to the reference sequence at this red locus. So if we pump this sample through the same pipeline, we would generate a read, then we would map it to the reference, and we'd hope that this is the mapping we get. This is the mapping where all of the bases in the read are aligned to their homologous base in the reference sequence. But this is the mapping we would actually tend to get, which is to say we wouldn't. Most mapping algorithms would return sequences that have diverged to that extent as unmappable, or else produce uninformative or nonsensical alignments. So we end up in this situation where we're systematically blind to some parts of the sample's genome, not because there's anything wrong with the sequencing data, but because the reference structure does not represent the sample's genome well at this locus. So this creates a number of biases, including some really nasty things like potential ethnic biases in medical research. So that's the bad news. The good news is that we actually know a lot about what kind of variation we would expect to see, at least at high frequency. There's a number of large-scale projects that have cataloged genetic variation in humans. So there's a community of researchers, myself included, that are working to use this population information to mitigate the biases that we have from interpreting sequencing data through the lens of a reference structure. And the way we do that is by incorporating the genetic variation found in the population directly into the reference itself. And when we do that, instead of being naturally represented as a string, the reference structure becomes very naturally represented as a mathematical graph. And I have an example here where each of the nodes are labeled with a DNA sequence. And then you can spell longer DNA sequences, for instance, an entire chromosome, by concatenating the sequence labels along a directed walk through the graph. And that gives you sort of a language for describing genetic variation like this SNP, or an indel, or a copy number variant, or the inversion from the example that I showed you a few slides ago. Now, the challenge with working with graph-based reference structures is that the genomics ecosystem is really wedded both conceptually and through software to this formalism of a linear reference. So when you are using graph-based references, you tend to need to do a bit of trailblazing to do even tasks that have well-posed solutions on linear references. And in that spirit, I'm now going to shift to talking about population genetic models, maybe a little abruptly, but I will dovetail these two subjects in the next section. So in population genetics, there's a very long tradition of probabilistic models of populations. Most of it derives in some manner from the Wright-Fisher model shown on the left, which describes inheritance moving forward in time as a random process. The more modern approach, referred to as coalescent theory, is to take the perspective of just the current generation, the current extant generation, which happens to be the generation that you can actually collect data on, in which case the same probabilistic model becomes a model of ancestry or a model of trees rather than a model of inheritance. In either case, those are genetic models. They are concerned with single loci, so they are not really genomic models. To move to genomics, you use this thing called the ancestral recombination graph, which deals with the fact that different parts of a chromosome can have different ancestral histories due to the complications introduced by recombination. So that's an important theoretical tool, theoretical concept. The ancestral recombination graph tends not to be used that often in practical applications. And the main reason, I think, is that it's just, it's a difficult structure to work with computationally. There are a lot of things that we'd want to use, or that we'd want to be able to compute for statistical inference on the ancestral recombination graph that are computationally intractable. So what we tend to use instead are more tractable approximations to the ancestral recombination graph, the most prominent of which is probably this approximation by Lee and Stevens. And the main simplification that they make is they assume we have a set of previously observed haplotypes that we treat in some abstract sense as the population. So here I'm depicting the haplotypes as rows in this binary matrix, where the columns are variant loci. And these are biallelic loci, so we code the two alleles as a 0 or a 1, and then relative to a linear reference, this completely determines a haplotype. The Lee and Stevens model is a generative model. It describes how new haplotypes are formed, and the process that they are formed in the Lee and Stevens model is as a mosaic of these previously observed haplotypes. Okay, so that's a very nice model for linear references. It does not, however, generalize to graph-based reference structures. And the main reason being that it depends on this organization in that binary matrix. And when you express a haplotype relative to a graph, you can't count on them fitting into a matrix anymore. They can have much more complicated relationships to each other. So now I'm going to describe a generalization of the Lee and Stevens model. Like the Lee and Stevens model, I'm going to assume we have a set of previously observed haplotypes. Now these are encoded as these blue paths in a graph. And it's a generative model of other haplotypes, like this pink one. And I should mention that in a graph-based reference, the natural way of encoding a haplotype is as one of these directed blocks of the graph rather than as a list of variants. All right, also like the Lee and Stevens model, we're going to kind of tell a generative story where the new haplotype is constructed as a mosaic of these previously observed haplotypes. And this is sort of the intuitive, conceptual derivation of our model from the Lee and Stevens model. There's also an algebraic relationship between their likelihood functions that I'm not going to show, but if anyone is interested, it's spelled out in our bioRxiv preprint. Okay, and we have developed a novel algorithm for computing the likelihood in this model. It's a dynamic programming algorithm. So in kind of typical dynamic programming form, it will compute the likelihood for increasingly long prefixes of the path. So we'd start with computing the likelihood of just a single node, and then in each iteration, extend it until in the final iteration, we have the likelihood of the entire haplotype path. It depends on this data structure called the GPBWT, which was developed in our lab. It is hard to say, and also a bit too complicated to describe in detail in a talk like this. The main things that I think you should know about it is, first, it's a succinct data structure for representing a cohort of haplotype paths like the one that the Lee and Stevens model is based on. It also supports some efficient algorithms. The main one for our purposes today is that it can do subpath counts. So what I mean by that is if we have a shorter path that we're interested in, say this one, we can count how many times that occurs as a subpath of all of the cohort paths, and it can do that in a manner that has no dependence on the number of haplotypes that are embedded in the GPBWT. The algorithm we've developed is also kind of centered on this conceptual framework that we call the rectangular decomposition. And the rectangular decomposition is a way of splitting up these cohort paths. Oops, excuse me. It's a way of splitting up these cohort paths into subsets. And the subsets are based on where they converge with and diverge from the query haplotype path in pink. So I'll show you what I mean by that. We have the example here. This first set contains all of the cohort paths, and those are the ones that have converged by A and have not yet diverged at A. It's sort of the trivial case. I think the next one's a little more revealing. These are the cohort paths that have converged at this node A and have not yet diverged at B. And notice this thick blue band over here is no longer in the set. And that's because in between A and B, those diverge from the query path in pink and take this other node. But they also converge back with the query path over here. And so they join this second set of paths that converge at B and have not yet diverged at B. So the whole rectangular decomposition looks something like this. We have these rectangles that shrink going to the right whenever a path or set of paths diverge from the query path. And then whenever new paths converge with the query path, we get a new row in this, this structure. So we're not actually so concerned about which paths are in which sets. But it turns out to be all the information we need to describe the recombination structure of the cohort relative to a query path in the counts, the size of these rectangles. And that happens to be something that we can compute very efficiently with the GPBWT. So after we do that, we get an upper triangular matrix that looks something like this. And it's an upper triangular matrix that tends to be very sparse away from the diagonal. And the reason for that is that to have values far from the diagonal, you need a cohort path that matches the query path exactly for a very long distance. And the longer of a distance you consider, the less likely it is that that has happened. So that's sort of the input data to compute the likelihood function of our probability model. But the dynamic programming structure mirrors it in shape. Now, I'm not going to show you the dynamic programming recursions because they're actually pretty complicated and a little opaque, and they take 2 or 3 pages of algebraic and combinatorial manipulations to derive. So again, I will direct anyone who's interested in actually seeing the math to the preprint. Instead, I will skip ahead to some analysis. It turns out that the runtime of this algorithm is proportional to the number of cells in the rectangular decomposition. So since this is the upper triangle of a matrix, that means in the worst case, it can take time proportional to the length of the haplotype squared, which is pretty large. But in practice, we do much better than that. And the reason is that this matrix has these sparsity properties. We also have the property that the worst case runtime is completely independent of the size of the cohort, which is we sort of inherit from the GPBWT. And to our knowledge, this is the only model for computing Lee and Stevens likelihoods that does that, has this property without losing any resolution in terms of where recombinations can occur. Okay, so I want to show you some results using this algorithm. First, I have some runtime results. Here is the dependence of the runtime on the length of the haplotype. And I told you before that it could be quadratic in the worst case. But in practice, you see we actually get something that's much closer to linear. There's also a dependence on the size of the cohort. And I know I told you that in the worst case, there's no size dependence. And that's true. But we are not hitting the worst case. We're in this sub-worst case regime where there actually is some dependence on the size of the cohort of haplotypes, because all else equal, a larger cohort will tend to give you a less sparse matrix. But the good news is that the relationship is highly sublinear. You can see the slope curving down over there, and then on the log-log plot, the slope is much less than 1. So those are both results that show this could be an algorithm that's practical to use as a replacement for the Leeds-Stevens model. We also want to know, does the probability model make sense? Does it produce results that are sensible? So to evaluate that, we have this other experiment where we've computed the likelihood of 2 different sets of paths. One set of paths, the one shown in black, are the paths taken by reads from the 1,000 genomes sequencing data. And the 1,000 genome sequencing, the reads are sort of inherently phased, which means that the variants on them respect all of the linkage disequilibrium relationships in the population. The other set of paths are random walks through the graph reference. And so those have no knowledge of population structure. They don't respect LD. So we'd expect them to, in general, have much lower likelihoods. And that is the pattern that we see. The read likelihood is concentrated over here on the right side of the distribution. The random walk likelihood are much more left shifted. They also have these multimodal peaks that correspond to walks that require novel recombinations that have never been seen in the cohort. Okay, so where we're going with this is first we want to extend and use this model for this kind of applications that you typically use Lee and Stevens for instance, genotype imputation on some of the newer, larger cohorts being published, like that of the Haplotype Reference Consortium, which is an order of magnitude larger than the data that we're using for these experiments, which came from the 1,000 genomes. And in those larger data sets, we think there's even more of an impetus for the favorable scaling properties of our algorithm. We also want to do a full generalization of Lee and Stevens. I did a bit of a sleight of hand, and the results I showed you were for a version of the Lee and Stevens algorithm that does not include mutations. The classical Lee and Stevens algorithm also includes both mutations and recombinations. We know how to do that mathematically, but we have not yet implemented it. And finally, kind of our end goal for this whole process is that our research group is working on a new form of genome inference that incorporates known population information kind of at every stage of the analysis, starting with the reference structure, but then moving on through later steps of the statistical processing. Okay, and so I would like to thank especially my co-author, Yohei Rosen, who couldn't be here, also Adam and Wolfgang from our lab, Eric Garrison from the Richard Durbin's lab, all of whose tools we used for the analysis, and then my advisors, David Hausler and Benedict Payton, and our funding sources. And I'm happy to take questions.
Anybody have questions?
Hi, thanks for your talk. If I had instead an assembly of short reads and I wanted to compute a likelihood that assembly is correct, or at least similar to a pangenome, could I use your tool to align that assembly to the pangenome and compute its likelihood?
Okay, so you have, yeah, I think in principle you could do that. Yeah. You would need some way to align long paths to a graph-based reference, and that's something that I think is still an open research area. We don't quite have like the same aligning, the same ecosystem of aligning tools that a linear reference has. But the probability model completely generalizes to that.
Okay. Thank you.
Yeah.
Thank you. In the interest of time, thank the speaker again.